Welcome back to another deep dive. It is great to have you with us today. We are starting off with a question that, honestly, it might sound a bit like science fiction at first.
It really does. Yeah.
Right. But I promise you, it is very, very real. So think about this. What if, instead of using toxic chemicals to poison cancer cells or, radiation to burn them away, we could just convince those cells to kind of grow up and retire?
It sounds almost too simple, doesn't it? Just tell the cancer to stop. But that is exactly the paradigm shift we are exploring today. We're looking at a specific cancer called medulloblastoma.
And just for some context for you listening, this is the most common malignant brain tumor found in children, right?
Exactly. It originates in the cerebellum. That's the part of the brain sitting right at the back of your head, the part that coordinates your movement and balance. And historically, the standard treatments for this are, well, they are incredibly harsh.
Yeah, we're talking about really invasive surgery, intense radiation, heavy chemotherapy.
Right. And while those treatments absolutely can save lives, they often leave these young developing patients with devastating long-term side effects. We see severe cognitive deficits, motor issues, and even lifelong endocrine disorders. Essentially, we are poisoning the developing brain in order to save it. But today, we are looking at an entirely different approach, something called differentiation therapy.
And to do that, we are diving into a truly groundbreaking 2020 paper. This was published in the journal Cell Reports by Cheng and an extensive team of researchers. The paper is titled, NeuroD1 Dictates Tumor Cell Differentiation in Medulloblastoma.
Which is a very heavy academic title. But what this paper actually does is fundamentally challenge how we view solid tumors.
Wow.
We are going to walk you through how researchers used incredibly advanced sequencing technology to uncover basically a secret population of retired tumor cells just hiding in plain sight.
Hiding right there in the tumor. And we'll look at a genetic switch called NeuroD1 that controls this retirement. And finally, we'll look at how a new class of epigenetic drugs might actually be able to force aggressive, drug-resistant cancer cells to permanently clock out. Okay, let's unpack this. Our first stop on this journey is understanding the secret life of these medulloblastoma cells. Where are they even coming from, and how did the researchers find out what they were really up to?
Well, to understand the cancer, you really have to understand the normal, healthy cell it came from. Medulloblastoma, specifically a major subtype called the hedgehog subgroup, originates from cells known as cerebellar granule neuron precursors.
Cerebellar granule neuron precursors, that is a mouthful. Let's just call them GMPs for short.
GMPs works perfectly. So in a healthy, developing human brain, these GMPs multiply rapidly, but only for a very short, specific window of time to build the physical architecture of the cerebellum. And then, crucially, they stop. They differentiate.
Okay, so they differentiate, meaning they mature into fully functioning, quiet, highly specialized neurons.
Precisely. They stop dividing and they start doing their actual mature job.
So if I'm picturing this for you listening, is it kind of like a bunch of incredibly ambitious college students? Like usually they go to school, they party for a few years, they multiply their social circles, but eventually they have to graduate. And that graduation event is cellular differentiation. They get a job as mature neurons and they settle down.
That's a really great way to look at it. But in this specific type of medulloblastoma, a genetic mutation occurs. Most often, it's a deficiency in a gene called PTCH1.
And this mutation acts like a dropout notice.
Exactly like a dropout notice. It breaks the brakes on a specific communication line called the hedgehog pathway. Because those brakes are broken, the cells are constantly receiving a chemical signal telling them to divide.
So in the college analogy, they drop out of school and just keep partying. They refuse to graduate, they multiply endlessly, and that uncontrolled division is what literally forms a physical tumor.
Right. But here's where the researchers made a stunning discovery. They wanted to know if absolutely every single cell in that tumor was partying, or if some were doing something else. To figure this out, they used a technique called single-cell RNA sequencing on mouse models of this brain tumor.
And for you listening, you can think of single-cell sequencing as an incredibly precise molecular sorting hat.
It really is a massive leap in technology. Previously, we only had bulk sequencing. If we use a different analogy, bulk sequencing is like taking a whole tumor, grinding it up, and looking at the average genetic signal.
Like standing outside a football stadium and just listening to the crowd roar. You know it's loud, but you don't know what anyone is actually saying.
Yes, exactly. Single-cell sequencing lets researchers isolate over 9,000 individual cells from this tumor, 9,164 to be exact, and read the specific genetic instruction manual each single cell is actively using at that exact moment. It lets you interview every single person in the stadium.
And when they conducted those thousands of microscopic interviews, the data revealed something totally unexpected. The tumor wasn't just a uniform, solid mass of dividing cells. It actually separated out into 3 distinct buckets.
3 very different states of being.
Right. So the first bucket was the dividing cells. These made up about 30% of the tumor, and they were highly expressing rapid cell division genes, things like CDK1 and CDR2. The second bucket, which was actually the largest at 41%, were quiescent cells.
Quiescent meaning dormant. They weren't actively dividing, but they still held all that dangerous mutated potential, just sleeping giants.
But the researchers were most interested in the third bucket. The remaining 22% of the tumor cells were fully differentiated.
Which was the shocker. When they looked at their RNA, these cells were expressing genes normally found only in mature, healthy neurons. Genes with names like TAG1, also known as CTN2, and TUB3.
So somehow, right in the middle of this aggressive, rapidly expanding tumor, nearly a quarter of the cells had spontaneously decided to graduate and settle down.
Which is wild. But seeing it on a computer screen in the sequencing data is one thing. You have to prove how these cells actually behave in a living organism. Clinical evidence is what matters.
So they took these cells and put them into mice, right?
Yes. They set up a fascinating case study. They took the tumors and physically separated the cells. They put the dividing, aggressive TAG1 negative cells into one group, and the 22% of mature, differentiated TAG1 positive cells into another group.
The graduates versus the dropouts.
Right. They then transplanted these 2 different populations of cells into the brains of completely healthy mice to see what would happen.
And the results here were totally black and white.
Right.
When they injected the dividing non-differentiated cells into the mice, it caused deadly brain tumors with 100% penetrance. Every single mouse got cancer.
But the mice that received the differentiated cells, 0 tumors. Nothing. Those cells had permanently lost their tumorigenic capacity. Even though they were born from a deadly cancer, they were completely harmless.
Which provides concrete proof that a fraction of cancer cells are naturally retiring on their own.
And this raises a crucial question for you to ponder as we move forward. If 22% of tumor cells naturally lose their cancer status, what is stopping the other 78% from doing the exact same thing?
Okay, before we move to the next layer and answer that, let's lock in what we've learned. Let's do a quick recap.
Sounds good.
Bullet 1, medulloblastoma, is a childhood brain cancer born from precursor cells that refuse to mature. Bullet 2, single-cell sequencing revealed that about a fifth of these tumor cells actually do spontaneously mature or differentiate.
Right.
And bullet 3, once differentiated, these cells permanently lose their ability to form tumors. They behave just like normal, harmless cells.
Spot on. So now that we know these cells have the capacity to retire, we need to find the trigger. What actually pushes a cell out of that endless division cycle and forces it into a mature state?
The researchers traced this back to a master switch called NeuroD1. Biologically, NeuroD1 is a helix-loop-helix transcription factor.
And a transcription factor's entire job is to bind to the DNA and orchestrate a massive cascade of gene expression that changes the cell's identity.
Wait, so if it's a transcription factor dictating maturity, is NeuroD1 basically the physical diploma in our college analogy? When you hand a cell this NeuroD1 diploma, it stops the endless partying, stops dividing, and actually starts building the structural branches of a normal neural cell.
That is a perfect parallel. To test the power of this NeuroD1 switch, the researchers needed to artificially flip it on in the cancer cells. To do this, they used a lentivirus.
And you can picture a lentivirus as a microscopic Trojan horse.
Right. They hollowed out a virus, packed it with the instructions for NeuroD1, and used it to deliver that code directly into the aggressive dividing medulloblastoma cells in the lab.
The outcome was profound. The moment NeuroD1 was expressed, cell division completely crashed. They measured this by looking at BRDU, which is a marker for active DNA synthesis, and those levels just plummeted.
At the exact same time, the cells started expressing MAP2, which is a marker for fully mature neurons. And just like the naturally differentiated cells we talked about earlier, when they put these supercharged NeuroD1 cells into mice, absolutely no tumors formed.
But here is where my mind gets completely blown by this paper. You have these cells. They have a massive genetic mutation, that broken PITE1 gene that is constantly screaming at them to multiply. The hedgehog pathway is stuck in the on position. But when NeuroD1 is introduced, they just stop. How is that possible if the DNA is still broken?
What's fascinating here is that the act of differentiation literally overrides the underlying genetic mutation.
Overrides it completely.
Yes, the cells still have the broken hedgehog pathway, the DNA is still mutated, but differentiation changes the cellular context so drastically that it chemically silences that pathway. The broken alarm system is still there, but the cell unplugs the speaker.
That is incredible.
It proves that cancer isn't just about a damaged genome. It is highly dependent on what the cell is actually doing with that genome. If you change the cell's core identity from a replicating precursor to a mature neuron, the mutation becomes totally irrelevant. The cell no longer has the machinery to even respond to the multiply signal.
That is such a massive shift in how we think about the disease. The cell's new identity just unplugs the broken alarm. So NeuroD1 is the key. It forces the tumor cells to retire, completely overriding their cancerous DNA. Which brings us to another huge question. If NeuroD1 is the ultimate cure, the perfect adulting switch, why is it turned off in the vast majority of these brain tumor cells in the first place?
Before we answer that, let's pause and consolidate this second major breakthrough.
You got it. So let's do our second recap to lock this in. Bullet 1, NeuroD1 is the genetic master switch driving tumor cell retirement. Bullet 2, forcing NeuroD1 expression completely stops tumor growth. And bullet 3, the act of differentiation is so powerful it overrides the cancer's underlying genetic mutations.
So we return to the mystery. Why is NeuroD1 turned off in 78% of the tumor? To figure this out, we have to step out of the realm of pure genetics and step into the realm of epigenetics.
Epigenetics being the incredibly complex system of chemical tags that sit on top of your DNA. These tags tell the cell which genes to turn on and which ones to keep securely shut.
Exactly. The researchers discovered that the NeuroD1 gene isn't missing from these cancer cells, and it isn't mutated either. It is simply being violently silenced by an epigenetic villain, an enzyme called EZH2.
EZH2. And this is such an elegant mechanism to visualize. Imagine the NeuroD1 gene is a beautifully written, step-by-step instruction manual for how to be a normal, healthy brain cell. The enzyme EZH2 is a strict, heavily armed prison warden.
A warden whose specific job is as a methyltransferase. It adds a chemical tag called H3K27E3 to the DNA.
Right, H3K27E3. Let's just call that the padlock. What this warden does is slap this massive, heavy chemical padlock onto the instruction manual. Because that padlock is physically blocking the pages, the cell cannot open the manual. It can't read the instructions for growing up, so it is stuck in its default state, which is blindly multiplying.
To prove this warden was responsible, the researchers used an assay called ChIP-PCR, chromatin immunoprecipitation. Think of this as molecular forensics. They are basically dusting the genome for fingerprints.
Looking for the padlock.
Yes, looking for that specific H3K27E3 tag. And sure enough, in the aggressive dividing tumor cells, they found those heavy padlocks plastered all over the promoter region, basically the start here tab of the NeuroD1 gene.
So EZH2 had completely locked down the start tab.
Completely. But the researchers didn't stop at just identifying the padlock. They wanted to see what happens if you remove the warden entirely. They used a genetic tool called shRNA to essentially knock out the EZH2 enzyme in the cells.
And the forensic evidence changed immediately. Without EZH2 patrolling the genome, those chemical padlocks vanished. Without the padlocks, the cell could finally read the manual. NeuroD1 levels violently spiked upward, and as a direct result, cell proliferation plummeted.
The warden is gone, the manual opens, the cell reads it, and it finally retires. It is a beautiful biological sequence.
So EZH2 locks up the NeuroD1 gene, which keeps the cancer aggressive, and that leads to the next logical question for you to chew on as you listen. If an enzyme is locking away the cure, could we just design a drug to break the lock?
And that is exactly the translational leap the researchers took. If EZH2 is the core problem, then an EZH2 inhibitor drug is the clinical solution.
Here's where it gets really interesting. We are talking about finding a chemical lockpick. The research team took a massive library of 244 different epigenetic compounds. They tested every single one of them on these tumor cells in a lab to see which ones could stop the rapid division and trigger the appearance of mature neuronal markers.
It was an incredibly rigorous screening process, but 3 clear winner drugs emerged. EPZ-6438, UNC-1999, and GSK-126. All 3 were specific EZH2 inhibitors. In the Petri dish, these drugs successfully stopped the cancer cells from dividing and pushed them right into that differentiated state.
But as we know, the real test of any therapy is always in a living, breathing system.
Right. So they took these specific inhibitor drugs and fed them to mice that were actively growing aggressive Peach1 mutated medulloblastomas.
And the data from these live animal models is just staggering. When the mice received these EZH2 inhibitors, their brain tumors shrank significantly. And when the researchers looked inside the shrinking tumors, they saw exactly what they had hoped for.
They looked at Ki67, which is the primary marker for rapid cell division, and it had plummeted. Meanwhile, Nuin, which is a marker for mature, fully differentiated, totally harmless neurons, spiked.
The drugs were working. They were successfully picking the lock inside a living brain.
But the researchers did one more experiment that I really want to slow down and emphasize because it is the ultimate proof of concept in this paper. They wanted to test these drugs against the absolute worst-case scenario. In the clinic today, we actually have an FDA-approved targeted chemotherapy drug for this specific hedgehog medulloblastoma. It's called vismodegib.
But cancers are incredibly adaptable. They mutate and they develop resistance to our drugs.
Exactly. So the researchers deliberately engineered a cancer cell with a new secondary mutation called SMOA1. This specific mutation makes the cancer cell completely 100% invincible to the standard targeted chemo, vismodegib.
Which is a total nightmare scenario for an oncologist.
Yeah.
You have a pediatric patient, the tumor comes back, and it is entirely immune to your best drug.
Precisely. It is a fortress wall that the chemo cannot breach. So they took these invincible drug-resistant cells and treated them with the new EZH2 inhibitors.
And the result? The EZH2 inhibitors completely suppress their growth. And the reason why is brilliant. These epigenetic drugs aren't attacking the mutated hedgehog pathway that the tumor built a wall around.
Right. They are bypassing that pathway entirely by attacking the epigenetic lock on NeuroD1. They force the cell to change its fundamental identity. And once the cell matures into a neuron, the resistance mechanisms don't matter anymore because neurons don't divide.
It's like the cancer spent all its energy building a massive reinforced fortress wall to keep the chemo out, and these epigenetic drugs just flew a helicopter right over the wall and landed in the courtyard. They bypass the resistance entirely.
It's a fantastic visual.
So to summarize this therapeutic breakthrough, EZH2 inhibitor drugs successfully remove the epigenetic padlock, forcing even the most violently drug-resistant cancer cells to mature and stop dividing forever.
Which brings us to a really profound question for you, our listener. This paper offers a stunning, undeniable proof of concept for differentiation therapy. How could teaching cancer cells to mature, rather than just trying to poison them to death, change the entire landscape of pediatric oncology?
It is incredibly hopeful. All right, we have covered a massive amount of complex ground today. Let's do a final sweep to tie all these threads together into the big picture.
Our final recap.
Yeah. Bullet 1, the enzyme EZH2 normally acts as a strict cellular warden, locking away the NeuroD1 retirement gene using heavy chemical tags, those H3K27E3 padlocks. Bullet 2, we now have specific drugs, EZH2 inhibitors, that act as molecular lockpicks, successfully removing these tags. And bullet 3, freeing NeuroD1 triggers aggressive, even drug-resistant tumor cells to differentiate, mature into harmless neurons, and permanently stop growing, offering a highly effective, vastly less toxic alternative to traditional chemo.
A completely new way to treat the disease.
So what does this all mean? Why does this specific 2020 paper matter so much in the grand scheme of medicine for you? It matters because it represents a massive paradigm shift in how we fundamentally understand cancer. For decades, we have treated cancer purely as a disease of broken, mutated DNA, a disease of rogue cells that must be burned or poisoned out of the body at all costs, even if it harms the patient.
Which is a war of attrition.
Exactly. But this research shows us that cancer is also an epigenetic disease of confused cellular identity. The cells haven't lost the actual instructions on how to behave. They've just lost access to them. We don't necessarily have to kill the cells. We can reteach them. We can give them their identity back.
If we connect this to the bigger picture, it challenges one of the most fundamental assumptions in biology. The researchers noted that oncogenic transformation, the actual process of a cell becoming cancerous, might actually be a reversible process.
Reversible?
Yes. Just think about the sheer magnitude of that statement. If aggressive brain tumor cells can be biochemically coaxed into becoming harmless functioning neurons, what other possibilities are out there? What other incurable, highly aggressive solid tumors, whether they are in the lungs, the liver, the pancreas, or the bones, are just waiting for the right molecular career counselor to help them grow up and retire? The code for a healthy cell is still there in the DNA. We just have to figure out how to unlock it.
It completely changes the battlefield. It turns a destructive war into a biological negotiation. And that is an incredibly exciting, humane frontier for science and medicine. We've given you a lot to process today. Keep pulling at these threads, keep questioning the old paradigms, keep exploring the incredible adaptable machinery of the human body, and we'll catch you on the next deep dive.